Hello guys, welcome to Future Doctor. This is Dr. Nawab Akhtar. Today topic is cell membrane. So see here parts of a cell. Here intermediate filaments, here ribosome, rough endoplasmic reticulum and nucleus. Here Golgi vesicles, Golgi apparatus. Here plasma membrane is present. Here mitochondria. Here lysosomes. These all are parts of a cell. So we will explain about cell membrane. Cell membrane is a fluid mosaic model, lipid protein model. Fluid components means lipid and mosaic model. Mosaic component means proteins and lipid present in bilayers. And this theory is given by Singer and Nicholson. See here two layers. This is phospholipid bilayer. Here protein channels, cholesterol, skeletal filaments present. Integral membrane protein is present. Here glycoprotein is present, glycolipid is present, and peripheral membrane protein is present. So cell membrane composition of cell membrane. First one is lipid, protein and carbohydrate. Major component is protein 55% and lipid 40%, carbohydrate is 5%. Firstly we will discuss about lipid. Three types: lipid plus phosphate, phospholipid, lipid plus carbohydrate, glycolipid, cholesterol. First we will explain about phospholipid. Five types. First one is phosphatidyl choline or lecithin. Second one is sphingomyelin. Third one is phosphatidyl serine. Fourth one is phosphatidyl inositol. And fifth one is cardiolipin. Phosphatidyl choline or lecithin, it is a major surfactant of lungs. Major surfactant of lungs. Component of lecithin is, major component of lecithin is DPPC, dipalmitidyl phosphatidyl choline. Deficiency of DPPC dipalmitidyl phosphatidyl choline leads to the RDS, respiratory distress syndrome or Hyaline-Membrane disease. Phosphatidyl choline or lecithin, it is a major surfactant of lungs. A major component of phosphatidyl choline is DPPC. If deficiency of DPPC cause RDS and Hyaline-Membrane disease. Second one is sphingomyelin. Sphingomyelin, it's also a minor surfactant of lungs. L upon S ratio, lecithin upon sphingomyelin, is greater than or equal to 2, it's represent the fetal lungs maturity. Means sphingomyelin is also responsible for assess the L upon S ratio is assess the fetal lungs maturity. L upon S ratio is greater than or equal to 2, it's represent the fetal lungs maturity. And sphingomyelin, here myelin means neuron, the sphingomyelin also present in neuron. And third one is phosphatidylserine. Cell membrane have two layers, outer layer and inner layer. Outer layer have lecithin and inner layer have serine. Means both layer have different phospholipid. It's represent the asymmetry of cell membrane. It reverse, serine is going to outer membrane and lecithin is going to inner membrane. Serine combined with annexin 5 and send to the signal. This signal name is eat me signal and causes apoptosis. Test for apoptosis. Annexin 5 stacking. And third one is phosphatidyl inositol. Phosphatidyl inositol, it is a IP3, act as a second messenger. Last one is cardiolipin. Cardio means heart. The cardiolipin present in mitochondria of heart. If anticardiolipin is formed in body, leads to the trypanosoma pallidum. Cause syphilis. Test for syphilis, VDRL test. First one is phospholipid is completed, second one is glycolipid. So we will explain glycolipid. Next one is glycolipid. Glycolipid is present in brain and GIT. It's two types. First one is gangliocytes and second one is cerebrocytes. Gangliocytes express in GIT. Express in GIT. Subtypes GM1 gangliocytes. Act as a receptor for cholera toxin. for a toxin. Toxin name is cholera toxin. This is all about next one is cholesterol. Cholesterol is present in between the phospholipid, and cholesterol is a precursor of skin. Precursor of skin. 7-dehydrocholesterol converted into vitamin D at the presence of sunlight, and also maintain the fluidity. So what is fluidity? Two things here. One is increase the fluidity, the second one is decrease the fluidity. So substance that increase the fluidity and substance that decrease the fluidity. Substance that increase the fluidity, it is a PUFA, polyunsaturated fatty acids. It increases fluidity. It is an essential amino acid. Example: linoleic acid, linolenic acid, and arachidonic acid. First one is linoleic acid, linolenic acid, and arachidonic acid. This is essential amino acid. It's increase the fluidity. And substance that decrease the fluidity, saturated fatty acids. Saturated fatty acid, example is stearic acid and palmitic acid. Stearic acid, palmitic acid. Here no role of cholesterol to maintain the to increase the fluidity and decrease the fluidity of cell membrane. It's only maintain the fluidity. So let's change the color. So cholesterol is a neither increases nor decreases fluidity. It's maintain the fluidity, called fluidity buffer. Thank you guys. See you next video. If you like the video, don't forget to subscribe the channel.